Welcome once more to this YouTube channel. Kindly subscribe to this channel. The subject area is new genetic technologies, a vital topic in medical genetics for biomedical postgraduate students, particularly the Ph.D. candidates. Now, what are the objectives? Candidates should know recent advances in genetics such as recombinant DNA, genetic engineering, and the Human Genome Project, and how they are applied in gene therapy, genetic testing, and the creation of genetically modified organisms. Candidates should also know the history of eugenics and its current manifestations. What are the specific topics? 1. Race and eugenics. They have to understand it from the historical perspective. That's they need to know race and eugenics from the historical perspective. They also need to understand reproductive choice and new eugenics. The second topic here is recent developments in genetic and genetic technology. Recent developments in genetics and genetic technology. A. Recombinant DNA. B. Genetic engineering. C. Genome sequencing. D. Gene therapy. E. Preimplantation genetic diagnosis. F. Genetic testing. G. Clustered regularly interspaced short palindromic repeats /Cas9. H. Polymerase chain reaction, that's PCR. I. Gene chips. J. Ancient DNA. K. Other recent developments such as protein structure solutions, quantum simulation, and precise genome manipulation. Right here is an illustration of clustered, regularly interspaced short palindromic repeats /Cas9. What you have illustrated there is a form of gene editing. So you have DNA, there's cutting of DNA so that you have regularly repeated sequences. And then these sequences are separated by spacers. So in the process, you are trying to manipulate the gene. You now have a desirable gene, and then you correct a disease process. So this is being applied now in the area of achieving a cure for sickle cell disease. Next, you have an illustration of polymerase chain reaction, that is PCR. You want to study DNA, like in genotyping, you want to know about doing PCR. So you get the DNA sequence of interest, the DNA segment, and then you amplify it into millions or even billions of copies, such that when the copies are so many, you'll be able to access the DNA and you understand it better. So you go through the naturation of the DNA strands, and then you annealing and extension. So at the end, you get to understand the genes, and it's applied in areas like genotyping. Then next, you have an illustration there of chips, what we call DNA chips array. What you have there is also, you are trying to assess the DNA, trying to study DNA, and then you have this DNA spotted on surfaces like glass surfaces or silicon surface. So you have the DNA sequences that are isolated. They are spotted on the glass sequence and the silicon surface, a glass surface or silicon surface. And this is applied in the area of, for example, you have the application in area of drug candidates, or in understanding a patient's susceptibility to certain disease condition. Do check out other videos on this channel for details of all these topics. Don't forget to subscribe to this channel. Thank you for listening. Goodbye for now.